Centrosome and Centrioles of Eukaryotic Cells by Dr. Rahman. History. Van Benden, 1887, first discovered the centrosome. T. Boveri, 1888, coined the term centrosome. Occurrence. Centrosome mostly present in all animal cells, motile cells of many green algae, bryophytes, pteridophytes and cyclophytes. Centrosome is absent in prokaryotic cells, amoebae, some algae, higher fungi, higher gymnosperms and in all angiosperms. Structure. Centrosome or cell center is a non-membranous organelle usually composed of a pair of centrioles, diplosome, embedded in amorphous paracentriolar materials called centrosphere. Around centrosphere are found rays radiating in all directions and called aster or astrosphere. Usually the two centrioles present at right angle to each other in inverted T or plus manner, but centrosome with multiple centrioles present in giant cells of both. Under optical microscope the centrioles appear as two-minute darkly stained granules. But, under M centrioles appear as two short hollow microcylinders with cartwheel-like organelles. A centriole is about 0.3 to 0.5 long and 0.15 in diameter. Each centriole consists of a whorl of 9 peripheral triplet fibrils tilted at an angle of 40 degrees. Each center is without consists of 3 microtubules or subfibers designated from inside to outside as A, B and C. The 3 microtubules are joined in such that the A subfiber is a complete microtubule having 13 protofilaments while B and C subfibers are incomplete and share 2 to 3 protofilaments with the A subfiber. The adjacent triplets are interconnected at intervals by Proteus CA linkers. The center of the centriole has a Proteus rod called hub, 2.5 nanometers diameter, from which a Proteus spokes radiate and connect with the subfibers. In between X thickenings, Y thickening present which connect both to X thickening and CA linkers by connector. The centrioles are surrounded by one or more series of dense, amorphous, protoplasmic spheres called micelles or paracentriolar satellites. Their position changes with the different stages of cell. Functions. New centrioles arise from the micelles, paracentriolar satellites, of pre-existing centrioles in the G2 phase of cell cycle. Therefore, centrioles are called self-replicating. They help in the organization of spindle fibers and astral rays during cell division. Therefore, they are called microtubule organizing centers, MTOC. However, the centrioles are not essential for this purpose. The cells of higher plants lack centrioles and still form a spindle. The plants contain motor proteins for this purpose. Though centrioles have not been found to contain DNA, yet they are capable of forming new centrioles with the help of micelles which function as nucleating. Centrioles help in cell division by forming microtubule organizing centers, MTOCs. Out of the 2 centrioles in a spermatozoon, the distal one forms axial filament or tail. Centrioles can be transformed into basal bodies. They act as basal bodies at the base of cilia and flagella. The distal centriole of a sperm gives rise to the axonema or axial filament of sperm tail. Basal bodies formed from centrioles give rise to cilia and flagella. Thank you. Please subscribe to Dr. Rahman YouTube channel if you like.